Hello, my name is Douglas Shepard. I'm an assistant professor in the Center for Biological Physics in the Department of Physics at Arizona State University. I wanted to give you a brief overview of the research taking place in my group. Behind me, you can see a fluorescence microscopy image. And what we have here are cells. The nuclei of those cells are labeled in magenta. And then we actually have individual RNA molecules. You may have heard about RNA more recently, either through your classes or in the news for the SARS-CoV-2 vaccine. And those two different types of RNA molecules are represented by orange and green dots here. And those images are imaged at roughly the hundreds of nanometer scale. So overall, if I had to sum up our research goals, it's that we're interested in building novel microscopes and fluorescence microscopy methods to then try and probe the way that molecular biology is regulated from a physics first perspective. So we can build this up in lots of different ways. We can start at the individual molecule level, or we can start at the cellular level. And so what we have here is a movie of rat lung. And so inside this tissue, we've labeled connective tissue in cyan and then different cell types in the other colors. So we can actually build up these large, three-dimensional data sets that can also have time components and then use these dense data sets to try and build a quantitative understanding of the regulatory mechanisms in biology. So this leads to a feedback loop where we generate microscopy data. We then posit some sort of mathematical model that's based on the physics. At its simplest, you can imagine two molecules are floating around in solution and they may eventually bump into each other. This can be represented by diffusion, which you can then model as a random walk. You can then use that model to try and fit and predict the observed behavior that you see. You compare that with advanced image analysis, and then you repeat this cycle until you're confident that you have a model that is predictive of the behavior that you're interested in, but again, taking the physical principles into account of what's happening inside of it. So to generate the data, we build what are called bespoke or custom microscopes, where we build them for their application. And really, in particular, my group works in two areas on this. We do what's called sort of light engineering, where we use the idea that light is a wave, and we use our understanding of optics to try and generate new types of microscopes. So on the left, I'll replay this movie. You can see that the laser light that is going into this little dish is actually changing. It looks like it's rotating. And then if we look on the wall, we can see what looks like a diffraction pattern. So it turns out what we're doing is we're actually interfering two different laser beams inside the sample to generate a pattern. We then use that pattern to probe higher spatial frequency features than a microscope normally see. This is so-called super resolution microscopy. This particular method is known as structured illumination microscopy. This allows us to build high spatial temporal sort of resolution movies of molecular biology in action. And so in this case here, we have a cell, and inside the cell, we've labeled the lysosomes, the mitochondria, and the cytoskeleton. So these are three key components of cellular regulation. We also can engineer the emission light. And so this is the light that comes out of whatever your microscope's doing. And so in this case, we use a method known as perfect remote focusing. What that allows us to do is build a three-dimensional imaging microscope. So here you can see we actually have a set of individual molecules that we're imaging in 3D, but we can do this with just one objective at the sample very rapidly for a method known as single objective light sheet microscopy or oblique plane microscopy. Using this, we can do extremely fast volume imaging. So what I have on the right is a flagella. So this is a small filament that's helical in nature. This is prepared by Dr. Rizal Harardi's lab here at Arizona State University, and this works in collaboration with them, where that flagella is actually rotating and tumbling inside solution, and we're imaging at roughly 30 volumes, so 30 full 3D volumes a second at the hundreds of nanometer scale. And you can see we can actually see this thing rotate and tumble in 3D. So all of this work relies on really understanding the wave nature and the physics nature of light and diffraction and all of these things, and then we build that up into a usable microscope. So a main project in the lab right now is something known as spatial transcriptomics. And so the thing we're doing here is we're doing extremely high throughput 3D single molecule imaging. So here we have an overview of a piece of human lung tissue. Here is a movie of that human lung tissue being imaged by the single objective light sheet microscope that I mentioned. And so you can see the tissue is flying by, and there's all sorts of little dots inside of it. Each of those individual dots is one RNA molecule. We then write custom localization algorithms, so things to find those dots on the graphics card processor. And we do this in a way that accounts for the physics. And so here you can see the actual data. And then here you can see the theoretical molecule fit. We can then build this up using biochemistry and fluidics and generate a map of, let's say, 14 different RNA species that are mapped inside of a piece of this tissue. And just to give you a sense of scale, the whole piece of tissue here, there's a 250 micron scale bar, so this is centimeter scale, and then here's a zoom in of one area. So this is a huge project we're working on in the lab at the moment, which is accelerating 3D molecule imaging so that we can use it for these applications. So just to make sure that you had the information if you're interested, our group website has been on all of the slides. We also have a GitHub. We do all of our work openly. Here's my name and my email address. We do this work in collaboration with a number of universities, and also we have a number of funding agencies that fund the work. Please feel free to get a hold of me and visit the websites for more information. Thank you.